All right.
So I will open this lecture first, Bob, by giving some announcement, and then I will introduce you, and then you can start the lecture after that.
Perfect. Okay.
Udah boleh mulai, Bu Evi?
Yes, okay.
Bisa? Okay. Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. Semoga semua dalam kondisi sehat. Hope everybody is in a good condition and healthy. Today, we have the 5th guest lecture program, and it will be delivered by Professor Gilbertson from UC Davis. Before we start with the presentation, I would like to remind you all students that we will have a question and answer session at the end of the presentation. So you can write your question and post it in the chat room, or you can raise your hand during the discussion, and then you can deliver your question directly to Dr. Bob. And please, during the presentation, turn off your speaker, and you can turn it on back again during the discussion. All right, let me now introduce our speaker by reading his CV. Here it is, Professor Robert Gilbertson, or I'd like to call him Dr. Bob for the famous one. So Dr. Bob has a very strong background education in biology and of course plant pathology from the University of Massachusetts and Colorado State University. And if I have no mistaken, Dr. Bob is one of Agrios students, right, Bob? When Dr. Agrios teaching in Massachusetts. Please correct me if I'm wrong. And Dr. Bob has a lot of research experiences. I think one page is not enough to list his research project, but I know he mainly works on plant virology and molecular genetics, with his favorite virus group, of course, Gemini virus, potivirus, and probably toscovirus. So without any further ado, I would like to welcome Dr. Bob. Please, Bob.
Okay. All right. Can everybody hear me? Okay.
Yes.
Okay. All right. Okay. Well, thank you, Asti, for that very wonderful introduction. And thank you for the invitation to give this talk today. It really is a great honor and pleasure for me, okay? Because as I'll mention, we have a very special relationship, okay? So, all right. So today I want to give you some background information and talk about some aspects of plant disease diagnosis, because I think it's very important to discuss that, particularly in the context of disease management, and then talk about some of the new technologies out there very quickly, and give you a couple of examples, an old example and a newer example of integrated management of virus diseases in particular. All right, but first, why don't I introduce you to where I'm from, okay? So where is the University of California, Davis? All right, well, let's see here. Let's see if I can get my laser. Okay, so obviously we're going to be talking about the United States. Okay, just to let you know, I do know where Indonesia is. I've never been there, but I know where to find it on the map. And as we were talking, I hope to visit someday. So we're in the Western United States, and then we are in the state of California. Okay, so here's California right here. You can see a blow up here. I'm located in Davis, California, which is right near the capital city of the state of California, Sacramento. Okay, now the state of California has a very nice university system that is composed of 10 campuses. UC Davis is one of those 10, and you can see here, this is where the UC Davis campus is located, right in the, next to the city of Davis in beautiful Northern California. Okay, so maybe it's not so beautiful these days. We have high rates of COVID. We had a lot of wildfires this year, but most of the time it's a very beautiful place to live. Okay, so all right, let's see. Oh, let's see here. All right. So how about the Department of Plant Pathology at UC Davis? Okay. So like I mentioned, there's 10 campuses that compose the University of California, and UC Davis is the main agricultural campus. So we are the primary campus that does agricultural work. University of California, Riverside also does a fair amount of agricultural research. And just to show you that agriculture is very important to us, our nickname is the UC Davis Aggies. Okay, so that tells you how important agriculture is. We're located in the College of Agriculture, and our College of Agriculture is one of the best colleges of ag in the world, in my opinion. We're located on campus at Hutchison and Robbins Hall, which is behind, Hutchison is behind here. And our department is consistently ranked as one of the top plant pathology departments in the country and in the world. Why? A couple of notable things. Last year, we hosted the International Gemini Virus Symposium on the Davis campus. And thank God we did that before COVID hit. We were able to get our conference in November 2019. We had a wonderful conference. The only bad thing was ASTI wasn't able to go. But we had a wonderful group of people attend. We had beautiful weather. And as I say, thank goodness we got it in before the COVID outbreaks. All right. Well, many of you may not know that ASTI and I share a common academic pedigree, okay? And in fact, we were both mentored by Professor Douglas P. Maxwell from the University of Wisconsin-Madison, and also Paul Ahlquist, okay? And ASTI and I were at UW-Madison around the same time for a few years, working in Dr. Maxwell's group, and working together on the same virus, actually, the very famous Pagomovirus bean dwarf mosaic, all right, BDMV, that ASTI got her master's degree on, and she determined the genome sequence and did a lot of nice work. So here is ASTI. Boy, ASTI, you haven't changed hardly at all. Being mentored by Dr. Maxwell here, we're having a conference. Here's Dr. Paul Ahlquist, one of the world's leading RNA virologists, Teresa Martinez from the Dominican Republic, me, ASTI, Josias Faria from Brazil, and Steve Hansen. So you can see it's an international group that we had even back in those days. So we gained a love for Gemini viruses and international agriculture. And now we've even continued our lineage with one of ASTI's students, Max Lee Wakabesi, coming to UC Davis to do his master's in my lab. So we're very pleased to have Max here, and he's doing a great job. All right, so I wanted to put this here just to remind me to tell you that I think plant diseases are one of the most fascinating and interesting subjects, and I'm always thankful every day that I went into this field, because after 30 years, I swear to you, I still get excited going into the field and looking at plant diseases, okay? So I know I was fortunate to find something that I really love to do as my life's work, and I'm still passionate about it. I go out and see these beautiful symptoms. It just still excites me today. The other thing about this is, and we were talking about this, I've been able to travel to many countries and see many different diseases in different types of situations. And that's been one of the real positive things, you know, about being in international agriculture and learning about these other diseases in those situations. And of course, diseases aren't static, right? Diseases are changing all the time. You've, I'm sure, heard about emerging and re-emerging diseases, and we're dealing with those in California and worldwide. We have sudden oak death, a very damaging disease of oak trees in California. Citrus greening is a huge problem, as I'm sure many of you know. It's devastating citrus production in Florida, in the US, and California is very worried. And we're very actively trying to keep this out of our state. Of course, whitefly transmitted Gemini viruses, thrips transmitted tospos, and then old diseases can come back and be a problem as well. So there's always going to be a need for plant pathologists, okay? Don't forget that, because the plant diseases, they're constantly changing and they're presenting new problems all the time. And one of the problems is, of course, how to identify them, because just looking at the symptoms is very difficult often to identify diseases. So it becomes very important to become good at disease diagnostics. This involves using reference books. It involves going out into the field and asking the right questions, using a combination of methods to do diagnostics, and then gaining experience. So whenever you have a chance to go out to the field with your professors, never miss that opportunity, because you're always going to be learning, you know, new things every time you go out to the field. Now, today, you can't do applied plant pathology and not do use molecular tools. In the old days, you know, applied was kind of like people who didn't do molecular, and basic was molecular. Well, today, even in applied plant pathology, you use molecular tools, especially for diagnostics and maybe even more things like looking at the genetic diversity and things like that. So everybody needs to know about molecular technologies, okay? You really do in today's world, okay? And I was fortunate, actually, when I got my PhD, I interviewed for a job, and a professor job, actually, believe it or not, at UC Berkeley. And I didn't get the job, and I was very fortunate that I didn't, because I went to UW-Madison for my postdoc, and I learned molecular biology, okay? And that was the best thing I ever did. So not getting that job was a good thing. So today, you need to know about serological diagnostic tests, nucleic acid-based ones, and these are, you know, standard tests that are used. So, you know, granted, in certain situations, it's harder to gain access to them, but still, these are the tools of choice today. You know, ELISA, I don't even use ELISA in my lab anymore. It can still be useful for high throughput, but I don't find it as useful. The Immunostrip tests, I hope you've all been exposed to those, these dipstick tests where you can get the results in 50 minutes. They're available for many of the most common viruses, but of course you need to know which likely virus or pathogen you may have to select the right one. But they're available for tobacco mosaic, cucumber mosaic, tomato spotted wilt, many of the common viruses. Not really, you know, there's one for Geminis, but I'm still not convinced it's very useful. But these are very good. And we use them a lot for these common viruses. They're easy to use, they're fast, you can use them in the field, and you don't need equipment, okay? You know, they're not cheap, but you don't need to use numbers of them. I mean, once you've identified what a symptom is caused by, you don't have to use a lot of them. Okay, and then of course, PCR, RT-PCR, I mean, conventional PCR, qPCR, you know, these are really for virus detection today. I'd say these are the methods of choice, really, that we're using. Now, whoops, let me just say, as many of you, I think, know, most viruses that infect plants, most types of them have RNA genomes, right? And, you know, and so to use PCR for an RNA genome, you have to convert it to a DNA copy, right? And so, you know, not, you know, and you know, now I would just quickly say to you, RT-PCR, of course, we have to do that because we have to convert those RNAs of those plant viruses into DNAs. But today now, everybody should know RT-PCR, right? I mean, this should be common language. You know, talking to growers sometimes in the, you know, their eyes would kind of glaze over. They would not, you know, if you start talking about reverse transcription PCR and things, they kind of tune you out. But today, everybody should know RT-PCR because they've been, if they've had a COVID test, they've had an RT-PCR test, right? Because you all know COVID is a big RNA virus. So to do a COVID test, you have to do an RT-PCR to be able to be tested, and it's actually an RT-qPCR. So now more of this technology should be common in people's vocabulary as we're dealing with this massive pandemic. And you as scientists who work on viruses should know all this stuff and be able to carry on a good conversation with an average person who doesn't exactly know what's going on with COVID, okay? So even though you're a plant virologist, you should be very interested in this and know what a messenger RNA vaccine is and why it isn't an attenuated strain of the virus. It's just a piece of the virus that you're putting into your body to make one specific protein. So, you know, be, you know, aware of this, all of this around us now, you know, with your plant virus knowledge to be able to talk COVID-19 virology too, okay? All right, so the other general concept I want to focus quickly on is integrated pest management, and you've probably all heard about it. It's a simple concept. You're just combining multiple strategies that will be either biological, cultural, genetic, chemical, into a package that you decide based on the knowledge of the biology of the pathogen. So you really have to know the pathogen well to come up with a good IPM strategy, okay? And the goal is to come up with an efficient strategy that minimizes the reliance on pesticides, okay? That's our long-term goal. Make it environmentally friendly, economically friendly too, because pesticides cost a lot of money. So there's 3 simple steps. Correct identification. That's very important, right? Because that's the only way to make the right choices that we're going to talk about in 1 minute. Okay, so you've got to understand the biology of your pathogen, the epidemiology of the disease, and in your area. And then you develop an IPM strategy based on the best strategies in your area for this type of pathogen. And what you were going to have here is a kind of Ipm Toolbox, as we say, based on your knowledge of the pathogen. You're going to select a range of you know, tools or, you know, components of your IPM strategy, of your IPM package. Now, everybody wants disease resistance, right? And it's typically conventional. Oh, you know, a lot of people don't like transgenics, right? You know, pathogen-free propagated material, that's always a very good one, okay? You've got to get out into the field and monitor. If you guys aren't going out to the field a lot, you are not doing the job right. You've got to be out in the field looking at your crop, looking, is it normal? Is there something showing up? Sometimes you have to use pesticides, no choice, okay? We're going to talk about crop-free periods in a minute. So there's a lot of options here, but you need to know which one's right for your pathogen and then which one's right for your type of grower, okay? Because if you're working with small growers, the type of IPM package is going to be very different than if you're working with big growers, right? Who have more mechanization and maybe more capacity, more money to do some of these other strategies, okay? Now, to make life even more difficult, we can add in insect-transmitted pathogens like viruses. So not only do you have to worry about the pathogen, the host, the environment, now you have to add an insect into it. So that's yet another complication, okay? And that can be very hard because these insects can be hard to work with. They can be hard to monitor. They can be hard to know when their peaks are, okay? And then we also have other issues with these viruses, symptomless hosts. Back to COVID-19. Are we hearing about symptomless hosts? Oh, yes. Well, symptomless hosts can be a big problem with plant diseases too, okay? So it's much harder to manage insect-transmitted viruses, you know, in general, and it's certainly just spraying insecticides doesn't work, okay? So you have to come up with more innovative ways. And even if you do that, these vectors develop resistance to the insecticide. So just spraying insecticides is not the answer. All right, so the answer, as I mentioned, is understanding the biology, knowing where the virus is overwintering, understanding the biology of the insect vector, and then trying to identify weak links in the pathogen disease cycle to target with your management strategy. All right, so I'm going to talk about two examples. There's an old one. Integrated pest management of tomato yellow leaf curl in the Dominican Republic, okay? And the other one is going to be a more modern, more recent one, is going to be IPM of tomato spotted wilt virus. All right, well, in the early 1990s, some very strange symptoms appeared on tomatoes in the Caribbean country of the Dominican Republic, shown right here. Okay, there's Florida. This is the Caribbean basin. Asti knows it well. Here's the Dominican Republic, okay? And this is a country of about 9 million people, okay? A very nice country, actually, very nice people. All right, well, they had a very nice little processing tomato industry on their island that allowed them to make enough tomato paste to provide for the population. And they like to eat tomato paste in the Dominican Republic. Well, in the early 1900s, they started to, 1990s, sorry, they started to see strange symptoms on their tomatoes that were associated with populations of whiteflies. And these tomatoes were stunted, the leaves curled upward, they turned yellows, especially in between, especially the veins. And basically they didn't produce any flowers because the flowers they produced just fell off and their yields were basically devastated. Looking at these symptoms, a number of people had commented that they looked like those of tomato yellow leaf curl, but tomato yellow leaf curl was only a disease known to occur in the old world. It was not known to occur in the Western Hemisphere where the Dominican Republic is. All right, well, these were pretty severe symptoms and they sure looked like tomato yellow leaf curl. If you had seen pictures of that, and Dr. Doug Maxwell at the time had been to Egypt and he was telling us, wow, that really looks like tomato yellow leaf curl, okay? I mean, really looks like tomato yellow. And it was devastating. This was a very susceptible variety that they were growing at the time. Napoli, this Italian open pollinated variety that, like we said, only had to smell TYLCV and it would get sick, okay? And the losses were staggering, okay? So our hypothesis was that maybe this was tomato yellow leaf curl. And by this time, the virus, tomato yellow leaf curl, had been completely sequenced. The genetic information or the genetic sequence had been determined. So our hypothesis was it was TYLCV. We designed a set of overlapping PCR primers based on the sequence of this TYLCV. And sure enough, when we used it with samples from the Dominican Republic, we amplified the expected size fragment. And then when those fragments were cut up with restriction enzymes, they had the exact same pattern as tomato yellow leaf curl from Israel. Okay. And then the sequence was determined 97% identical. Okay. So that's not good. So they have introduced the worst virus of tomato into the Dominican Republic. That was not a good thing. All right, so then the question became, how widespread was it? What other host was it infecting? And back in the day, and Asti will fondly remember this, we had developed this method called nucleic acid squash plot hybridization, where you could just squash samples from leaves of plants you wanted to test for a pathogen like tomato yellow leaf curl onto this nylon membrane, okay? And then you would denature the DNA on here. You would hybridize it with a radioactively labeled, in this case, tomato yellow leaf curl probe. You would let hybridization occur. You would wash the probe off, and only where there was TYLCV DNA would have you retained the radioactive probe. You put a piece of X-ray film over it. The next day you expose the X-ray film, and wherever the radioactive probe was, you get a dark spot that corresponds to any of the plant samples that were infected with TYLCV, okay? So we did this in the Dominican Republic. We took these and hybridized them to see which ones were infected with tomato yellow leaf curl virus, okay? And this just shows you some results. What we quickly found out was tomato yellow leaf curl was almost only in the tomatoes. It wasn't in weeds. Even if they had golden mosaic or mosaic symptoms, the weeds were pretty negative. Exception was Datura, which is a weed in the tomato family, but almost all the positives were only in tomato. So we told the government, why don't you, because tomato is the main host, why don't you use a tomato-free period where you try to break the cycle of the virus? Because if tomatoes aren't around, and we know whiteflies have about a 1-month life cycle, we might be able to break the cycle of the virus. And the government went ahead and imposed that. Not only did they impose a tomato-free period, they made it a 3-month whitefly host-free period that included beans, cucurbits, and peppers, okay? And this is what they go around posting. This is the announcement of this host-free period that will be carried out in the north and the south processing tomato areas of the country. So you can tell how bad this problem was for the government to be willing to do this, okay? But the government said, you need to provide us evidence that the host-free period will work. So we came up with the idea of doing PCR testing of the whiteflies at different periods of time at monthly intervals. And what our collaborators in the Dominican Republic would do would send us the whiteflies in alcohol to UC Davis, and we would then do a quick extraction and do PCR to detect the presence of the virus inside the whiteflies, okay? So as I mentioned, there was a northern production area, and there still is, and a southern production area. And these are the sites they collect the whiteflies, and then they send them to us every month, okay? And then we do the PCR, we run the PCR, and we look for a diagnostic. It's about 350 base pair fragment of TYLCV. So during the growing season in the north and the south, here's the TYLCV band that you get from the whiteflies. All of them virtually were loaded. If you do a southern blot, this confirms it's a TYLCV fragment. So during the growing season, the whiteflies are loaded with TYLCV, absolutely loaded. Tremendous titers, okay? Now, whoops. When we go to the host-free period, look at that. Hardly any of the whiteflies are positive. And so this is 2 months into the host-free period. Now, there's still a few weak positives of whiteflies, and these turned out to have come from, in one case, tomatoes where growers were cheating. Can you believe that? That somebody would try to be cheating during the host-free period? And another one, pumpkin here, so, and another tomato. But look at how dramatically, compared to during the growing season, everyone's positive. Host-free period, hardly any are positive. So the government had that evidence to say, yeah, the host-free period actually works. And you can plot this right along. You know, a lot of virus during the growing season, host-free period, it drops, growing season builds up, host-free period, it drops, and so on. So it worked very well. And as I mentioned, the government enforces it. It gives growers about a 4 to 8 week window before the virus comes in. Okay, it includes peppers, beans, and cucurbits. We have worked very hard, or the government has, and the companies there, to educate growers why this is so important, but violators continue to be a challenge. Well, the virus didn't disappear because I said you get a 4 to 8 week window. So eventually the virus comes back. And since there was not much in the whitefly, where does it come from? How does it survive? Well, there was still some in the whitefly. We don't have problems with volunteers because during that host-free period, it's dry and hot. Violators are a possibility, and then reservoir hosts. But as I mentioned, we didn't find any weeds that were positive with that squash blot method, okay? But we did observe that we often found the virus coming in first at the edges. There was an edge effect, okay? But all these weeds, they didn't have symptoms, or if they did, they were negative TY. So then we had the hypothesis that maybe there were symptomless weeds there. And so we went in and collected leaves from the predominant weeds that didn't have any symptoms. We did a PCR, we used higher amounts of DNA, okay? And we ran a longer program. And lo and behold, we started detecting the virus from symptomless weeds. It was confirmed as the TY fragment with the southern blot. So symptomless weeds were overwintering the virus. Now, probably they're not very efficient for the whitefly to acquire the virus because, again, remember, we get this 4 to 8 week window before the virus shows up. But this is probably the explanation of where it is. And this is one of Cleome. This is one of those weeds, and it's all over the place. So you aren't going to eradicate it. No way. Really, we think the virus overwinters in very low levels in these weeds. It takes a while for the whiteflies to, and plus you have a whitefly host-free period to build up, to get it out of these weeds, to put it into tomato, and then for it to get going in tomato. So that's how you get this beneficial period of host-free protection, okay? So now the IPM package in the Dominican Republic, today as we speak, in fact, we're still testing the whiteflies for them. A 3-month host-free period, mandatory as I mentioned. Early maturing hybrids planted right after the host-free period. Now many companies have developed TYLCB resistant varieties, so you can use them. They're not as high yielding as the early maturing hybrids, but they're very nice, okay? So there's good TY resistance out there in processing and fresh market tomato today, okay? The breeders have done a good job, all right? The selective use of insecticides based on monitoring populations. And then at the end of the growing season, you've got to use extensive sanitation combined with the host-free period, and then monitoring to make sure no changes have occurred. And that has really saved the industry in the Dominican Republic. And today their yields are far higher than they were before the virus was introduced because of using better hybrids, improved growing conditions, and so forth. So it was really a success story, but still one that you have to work very hard to make sure continues to be successful. All right, so now I want to switch gears and talk about tomato spotted wilt integrated pest management, okay? And this is a disease that's economically important in my state of California here in the USA, and it's particularly important in processing tomato varieties, okay? So in California, I'm not going to talk about this, but we grow about 35% of the world's processing tomatoes industry, okay? And these are the tomatoes that are, you may hear them called industrial tomatoes, that are used for canning and for making sauce and things like that. So it's a very important industry, over $1 billion in the state of California. But tomato spotted wilt, which is this disease right here, is a big problem for us, okay? And it can show up early where the plants get stunted and they turn yellow and almost die and almost look like disease because of the necrosis. During the growing season, you see this kind of bronzing and necrosis. Any of you who have seen tomato spotted wilt would recognize this necrosis and bronzing and yellowing is very, very distinctive. And then by the end of the season, when the fruits form, spotted wilt forms the very diagnostic ring spots and blotchy symptoms on the fruits. And these kind of strikes at the end of the plants, when the plants get infected late, the virus just goes to these shoots where the fruits are, and you see this type of symptom. So just for this one disease, See how challenging it is for diagnostics? Because it's very different depending on the stage of growth, all right? And again, this is the type of thing that you learn with experience of working with these diseases, okay? Because believe me, when you go out to the field and, there's a grower there, they're going to expect you to know what you're talking about. And they'll also start asking you all kinds of questions about other things. And you have to, look sort of intelligent, or at least say, I know someone you can talk to about that, okay? So believe me, you can't just go out to a field and say, oh, don't ask me any questions about anything other than tomato yellow leaf curl, because that's all I work on, okay? You're not going to look very good out there, and certainly not somebody who graduates from UC Davis. They better not go out there and say that, all right? So this is one of the challenges, okay? All right, so what about this tomato spotted wilt disease, all right? It's caused by the virus, tomato spotted wilt virus, okay? Now, as Asti told you, I love Gemini virus. They're nice circular single-stranded DNA viruses, and they're easy to work with. But sometimes a problem comes up and you have to work on it, whether you like it or not, because it's important for your growers. And I remember going down in 2007. I went down with. The University of California has a very nice extension system. I forgot to mention that. And we work with farm advisors who work in the major counties, and I'll show you these counties in a minute. And so I work very closely with a farm advisor named Tom Turini, and he's really good. And I remember we went down and we met a couple growers, big growers in Tom's area. And we were looking at spotted wilt in this grower's field, and it was pretty bad. And this grower looked at us and said, he pointed his finger at us. He said, you, talking to, UC Davis, need to solve this problem for us, okay? And, that's the type of thing you're going to hear sometimes. And, and of course, we're like, yeah, later down the line, of course, my rule of is something a big enough problem here is, will you fund any research? And they, to their credit, did fund a fair amount of research to learn about the biology of this virus in California, and then come up with IPM strategies for them, okay? So by 2005 in California, we were starting to lose a lot of money due to this virus. And growers do not like to lose a lot of money, okay? They get very unhappy, and I don't blame them, okay? So we developed a big collaboration. It's probably the closest thing to the coronavirus that we have, because it's covered by a membrane, outer membrane, embedded with spike proteins, glycoproteins. Coronavirus, that, has a crown. That corona means a crown in Spanish, because of these glycoproteins that are on the outside of the virus particle, right? And you've all seen that, right? And in fact, those spike proteins are exactly what are being expressed in you to make the that the vaccine is, that then gets recognized by your immune system to make the antibodies to make you immune, hopefully, to COVID, okay? Well, these are the same spike proteins right here. And in fact, in this case, COVID has one big RNA, like 30,000 nucleotides. I mean, that's a humongous RNA. Okay, this one has 3 RNAs that are about 15 kb total. And they're all coded by a nucleocapsid protein, just like COVID is, all right? So it's a very complicated and difficult virus to work with, all right? It has a big genome, and these RNAs are negative sense, all right? So this is like the last virus in the world you'd want to work on, especially as a PhD student. If, although recently, infectious clones of this virus have been published, which was a huge breakthrough. And I don't know how easy they are to work with, but that was a big breakthrough, okay? So, transmitted by thrips, and here are the thrips, and a very important thing about thrips that you need to know if you know about these viruses, that for this adult thrip to be viruliferous, that is to be able to transmit the virus, it must acquire as these little instars or juveniles, okay? So these little guys need to feed on an infected plant for this adult to get the virus. And you should immediately think, oh, that gives you a delay early in the season because somehow this thing, a female has to lay eggs on an infected plant, and then these little guys have to come out and feed and acquire the virus, and then they become a pupae, and out of the pupae comes this adult, and only then can that adult transmit the virus, okay? So that's a very valuable little biological piece of information for you to, use in your IPM program, okay? And then the other thing is this virus is not seed transmitted in tomato. It's not transmitted by touch, by contact, and it doesn't get transmitted through the eggs of the thrips vector, which is really good, okay? So those are other biological facts that you need to know when you develop your IPM program. All right, so we developed an IPM program, okay? And I'm not going to go through all the details of it. We put it in a flyer for the growers. We give lots of talks. So, it turns out our transplants, we establish all processing tomato fields now in California with transplants, and they're produced in greenhouses. And we monitored them very carefully. They didn't have spotted wilt, and they had very low populations of thrips. So the problem wasn't coming from the transplant greenhouses, okay? Then, fortunately for us, right when we started our project, companies were coming out with resistant varieties. And what did I tell you was the best strategy you could pick if you could pick anyone? And this is what I tell my students in class. If you can't remember a management strategy for a disease on a quiz or a lab practical, guess resistant variety, okay? That's your best one to guess. But if you can pick, you always want a resistant variety, of course, as an option. Of course, it has to be a good variety that growers like, right? Well, fortunately, these came on board right around when we were developing our IPM strategy. So we said, yeah, that's one of the big things you want to use. And these are very good resistant varieties, as I'll mention in a minute about this gene. They don't get any symptoms, all right? And that's what growers like. All right. But when your resistance is that strong, what are you doing to the pathogen? Yeah, you're pushing on that pathogen, right? You're putting a lot of pressure on the pathogen to do what? To mutate. And what do RNA viruses do really well? They mutate, okay? Because they don't have any proofreading in their genetic information. And they don't care if a few mutants die. Nobody's shedding any tears over them because you're just looking to select for a winner, okay? And that's what happens out there. Okay, now, we also told our growers, get out there early and spray when the early generations are out to slow down the appearance of those viruliferous thrips, okay? And we tell them when that is using a degree day model that we've developed so we can predict when the generations show up. And then we work with our growers to rotate insecticides when they trigger their sprays for these early generations. Now, unfortunately, there are not really great insecticides out there for thrips, okay? If you know any of them, I'd love to hear about them. There are very good chemicals for whiteflies. There are very good chemicals for aphids. There are very good chemicals for leafhoppers. There are not good insecticides for thrips. So you're stuck with, a relatively few, the spinosids, radiant, things like that. There's a few others that can be used. Some organophosphates, dimethoate in California is commonly used. Lanate, methomyl, which is a very toxic one. I hate that one. But, there aren't a lot of good materials for thrips, and that's one of the possible viruses. For fresh market, small farmers, Rogan. Well, watch your plants. If you see a few plants with symptoms, pull them out as fast as possible, right? And that makes sense because I just told you that the viruliferous adults only come from infected plants. So by getting rid of the infected plants as quickly as you can, you again reduce the number of viruliferous adults that are the only ones that fly, okay? And then, plowing under the field, sanitation, getting rid of the old plants. You've got to do that because if old plants left in the field sprout out, they have tons of virus. So this was provided to our growers and in this pamphlet, and it worked wonderfully, okay? And, you know, we gave them this program, and really the resistant varieties were the most important thing, you know, to be honest. And, you know, they were quite happy. Okay. And so were we. All right. Because, you know, that's a win when the growers are happy. And this is just our degree day model that we have online, and growers can check to see when the second and third generations appear. My colleague Neil McRoberts writes blogs telling them what's going on every month and every 2 weeks in the early season to tell them what's going on, what generation is out there, and what our recommendations are. Okay. And then we've recently published this in a kind of nice publication that brings all of the research together. Well, that was very good. And we were patting each other on the back and, you know, so on and so forth. And you shouldn't be doing that against Mother Nature. Okay. That's a bad idea, because I've learned in 30 years, you don't mess with Mother Nature. Every time you think you know what's going on, Mother Nature throws you a curveball. At least that's been my experience. I've been humbled many times. And indeed, in 2016, my friend Tom Tarini gets these phone calls that fresh market fields with SW5 resistance are developing symptoms, regular bad symptoms of spotted wilt in Fresno County. Okay. So we immediately go down there, and sure enough, those are spotted wilt symptoms. I can assure you of that. They were immunostrip positive for TSWV, as I'll show you in a minute. And they were also RT-PCR positive. And then sequencing the fragment, it was spotted wilt, no question. Okay. And, you know, so we're like, uh-oh. Now, we had already known that resistance breaking strains of spotted wilt were a possibility. Okay, because they had been known in Europe, in Spain and Italy, using the same gene, the SW5B gene. They had selected for resistance breaking strains that were no longer controlled by that gene. Okay, and there was a motif, as I'll show you in a minute, in the movement protein gene that was a marker for this resistance, at least for the strains in Europe. Okay, so we already, you know, now here's another interesting thing. What do you tell the growers? All right. So when we developed the IPM program, the original IPM program, you know, I was telling them, now guys and girls, there are these resistance breaking strains. So we need to use the other strategies, keep the populations of thrips down, do the other things in addition to the resistant varieties, or else we're going to put so much pressure on the virus, we'll select for a resistance breaking strain. Now, you know, I wasn't trying to threaten them or anything like that. And I don't think they really paid much attention to it, because they really just relied on the resistant variety. Okay. All right. So now we have this situation. Is it RBTSWV? All right. So first, before I tell you the answer, I wanted to tell you a little bit something about this gene, this SW5 gene. Okay. It's a single dominant gene. It was introgressed from a wild species, like many of them are. It is a very typical, resistance gene, coil, NBS, LRR type. Max knows this very well. And as I mentioned, there's what's called an effector from the virus that's recognized by this resistance gene to trigger the resistance. But if a mutation occurs in this movement protein, which is made off this messenger RNA here, so that you get an amino acid change at position 118 or 120 to amino acid Y or N, those were the amino acids that the Europeans found could break this resistance gene. And sure enough, when we looked at the sequence, so we went ahead, we had a PCR test for the resistance gene itself. It was there. As I mentioned, positive with immunostrips, positive by RT-PCR. We amplified the movement protein gene sequence. We translated the amino acids. We looked at 118, look at that, a Y. The wild type should have a C there. So sure enough, our California resistance breaking strain had the Y118 that was characteristic of the resistance breaking strains in Europe. And we further confirmed that these resistance, we took one of the resistance breaking strains, we inoculated a whole bunch of SW5 varieties that were grown in California. And here you can see the RB strain here, the wild type strain. The RB strain infected every one of these so-called resistant varieties. And of course, both of them infected varieties that didn't have the SW5 gene, okay? So this new strain is breaking the resistance in everything. And we developed an RT-PCR test where we could detect it specifically. So we have specific primers for the RB versus the wild type strain. And now we've developed a LAMP assay also that can tell us if we have RB or wild type in less than 1 hour. Okay, so we can run these tests and tell a grower what they have. So now we're looking at where this RB is. These are the biggest tomato growing counties in California, and this red shows you where the RB strains are. So unfortunately, they're right where the biggest production is, and they've spread. They're the predominant strain by far now. These northern counties, we're starting to find it. We haven't found much yet, but we're very worried that it will get there soon. All right, so we have to convince growers to go back to the old IPM strategy more and be more vigorous about using the old approaches, okay? But what we really want to do is find a new source of resistance, right? But the problem is we've screened a lot of things from Europe even that supposedly are resistant to the European. These materials here were supposed to be resistant to the European RB strain. They're resistant to our wild type, but none of them are highly resistant to the California RB TSWV. So it's a monster. We haven't found resistance, although now I'm hearing some rumors that a couple companies might have some resistance. Now you can always go back to wild species, okay? But even this RB TSWV infects a lot of these wild species. But we did find a few here where there's some that may have some plants that didn't get infected. But man, if you go back to wild species and you try to make crosses, and I mean, I don't have time in my career to do that. You young people might, but not me, okay? So we have this monstrous resistance breaking spotted wilt that we have to get rid of, or we have to find some solution to. And this is just another example to you guys that these pathogens are not static, okay? They change, they move, they modify. So they're always on the move. And so things you have to always be looking on the lookout for introduction of new pathogens, new strains, and new, and things that are going to challenge your production systems. Okay, so that's it. I wanted to thank a few people in my group. Oscar Batuman, who really was the leader on doing the research on the spotted wilt project. He is now a professor at the University of Florida, ironically working on citrus screening. Tomas Melgarejo is a wonderful postdoc in my lab who I've been fortunate to have for, he did his PhD with me and then he came back to do a postdoc. See, Max, even Tomas came back. He's really great. Dr. Maria Rojas, who is the lab manager and a senior project scientist in the lab who has been responsible over the years for many projects. Asti knows Maria very well from the days in the Maxwell lab, which is where I met her as well. Dr. Monica Macedo did a lot of work on the spotted wilt characterization as well. Raquel Salati worked on the TYLCV project. Margaret Cespedes Kati is helping us on the spotted wilt. Neil McRoberts is my epidemiology colleague here at UC Davis, who helped develop the degree day model. Diane Ullman entomology professor here who I've worked with many years on tomato spotted wilt, and then my good friend Tom Tarini, the farm advisor in Fresno County, who I've had a long collaborative relationship with, and the California Tomato Research Institute, who's funded this. And I forgot to include my friends from the Dominican Republic, but they have also been very generous in supporting research on TYLCV over the years. And, you know, it's just a good example of how it's paid off many times. When I started working with that company, in this particular company, they were like the #4 tomato growing company in the Dominican Republic. Now, after investing in some research and knowing what to do, they're the #1 tomato producing company in the Dominican Republic now. So by investing some money in research, it always comes back to pay itself back multiple times over, you know, and you know, but you always have to get out there and remind growers of that. So it has been, again, my honor and pleasure to give you this talk today. I hope you found it interesting. I really appreciate ASTI inviting me to do this, because not only is it scientifically important, it is personally important, and perhaps even more so, as I fondly look back to the days in the Maxwell lab when ASTI and I were working on BDMV, and Dr. Maxwell came back to do a leave. You know, he had been the department head for a long time. I mean, he took a year off to come back and work in the lab and work quite a bit with ASTI. So I don't know, ASTI, Max may not want me to take a sabbatical and come work with him every day in the lab, but I know you, I know Dr. Maxwell enjoyed it immensely, definitely. So I'm going to go ahead and stop sharing my screen. And again, thank you so much for your attention and for allowing me to give you this talk today. It was a great pleasure. And if there's any questions, I'll be happy to answer them. I expect Max has at least one question. Right?
Okay. All right, Bob, thanks a lot. Very excited information and very nice lectures you gave us. Before we start with the questions session, can we take a pictures with you? Just for a documentation. Yeah, so yeah, the students have the documentation that they have the lecture from Dr. Bob.
Very good. Well, it is my pleasure talking to you students. Okay. Yeah. You know, and I want to encourage all of you, you know, if you find plant pathology to be your passion, like I have, to stay, you know, it's a wonderful field of science to go into. Okay. I love it because, you know, I've worked on bacteria, I've worked on fungi, I've worked on viruses, I've worked on viroids, ASTI. You know, so you know, I think it's really, you get to work on a wide range of diverse things. It's so interesting, and there's always something new, as I just told.
Okay. Sudah, Mbak Ana, siapa yang ambil pictures?
Lagi ya, Bu?
Yeah.
Udah?
Ini baru 25 orang ini yang menyalakan video.
Okay. You can turn on your video on.
Sorry, okay. Oh, okay. Can you see me?
Yes.
Okay.
Satu, oke. 1, 2, 3.
Okay.
Sudah, Bu Asti.
Oh, oke. Alright, thank you.
Mhm.
Baik, pertanyaan silahkan, saya lihat di chat room belum ada ya. Kalau tidak di chat room silahkan raise your hand and you can ask directly to Dr. Bob. Tidak usah ragu-ragu, don't be hesitate. Bob is a very nice guy and he will answer any kind of questions. Any
And I'll be
kind.
I'll be very disappointed if you don't have any questions.
That's
okay?
right.
It'll make me sad. You don't want to make me sad, right?
Yeah.
You know,
Okay.
I push my students to ask questions all the time, right, Max?
Right.
Oh, Max knows.
Do you
Or
want
Bu
to?
Asti just review a little bit. Bu Asti review the lecture. Could be someone couldn't catch it well.
Bahasa Indonesia, Bu.
Yes.
Okay. Okay, Bob, I will do a little, a short summary of your lectures in Bahasa Indonesia. So
No problem.
Alright, just be patient for a while. Oke, jadi mudah-mudahan tadi jelas ya yang disampaikan Dr. Bob. Bahasa Inggrisnya sangat jelas, kemudian penyampaiannya juga sangat bersemangat, jadi harusnya Anda bisa mudah menangkapnya. Jadi intinya ada dua permasalahan penyakit yang disampaikan. Yang pertama adalah penyakit pada tomat, tomato yellow leaf curl virus, terjadinya di Dominican Republic, ya, di negara Dominican Republic pada tanaman tomat. Kemudian yang kedua adalah penyakit yang disebabkan oleh tomato spotted wilt virus, ya, terjadi di California. Ya, jadi dua penyakit ini kebetulan virus dan punya vektor. Yang satu tadi vektornya whitefly, yang satu vektornya thrips. Nah, sebetulnya intinya dari materi kuliah ini adalah bagaimana kita mendesain IPM, mendesain Integrated Pest Management (PHT) itu ya, supaya bisa mengendalikan atau menurunkan insidensi penyakit ini. Dan semua berawal tadi ya dari identifikasi yang benar, kemudian mengetahui biology of the virus, tahu biologinya, epidemiologinya, host range, kemudian serangga vektornya. Nah, dari situ baru kita bisa menyusun strategi apa yang akan digunakan. Nah, untuk tomato yellow leaf curl virus, strategi yang dipilih itu adalah host free period. Artinya petani tidak diperbolehkan menanam tanaman yang menjadi inang dari virus atau dari vektor. Untuk kalau tidak salah tadi 2 bulan. 2 bulan. Jadi petani itu bisa serempak ya tidak menanam tomat gitu. Ya, di Dominican Republic itu bisa. Saya nggak tahu ya kalau di Indonesia bisa apa enggak tuh petani nggak boleh nanam gitu ya. Nah, tapi ternyata itu bisa menurunkan penyakit gitu. Ya, berhasil menurunkan penyakitnya. Nah, kemudian untuk tomato spotted wilt virus di California, mereka mencoba strategi mengembangkan varietas tahan, ya, varietas tomat yang tahan terhadap virus tersebut. Nah, tapi sayangnya setelah beberapa tahun, ini saya nggak tahu tadi juga nggak jelas ya berapa tahun, ketahanan itu patah. Ya, jadi tanaman yang tadinya tahan tiba-tiba terserang gitu. Dan ternyata kenapa terserang? Karena muncul new strain dari virus tersebut. Ada strain baru dari tomato spotted wilt virus itu yang mematahkan ketahanan tanaman tomat. Nah, sehingga mereka mencoba untuk mengembangkan varietas baru lagi, sambil selain itu mereka juga menerapkan prinsip-prinsip PHT yang lainnya. Saya kira itu inti dari presentasi Dr. Bob. Silakan kalau ada pertanyaan apa saja, nggak usah sungkan-sungkan. Okay. Max, maybe you want to start with the questions.
Okay, maybe I'll start with one question. So since the resistance genes in tomato doesn't work, as you explained, Professor Gilbertson, that it works, but then there was a strain that can break the resistance. What would be an alternative that we can use to maintain that this won't happen again if we found another resistance genes, or for the future? just in case it will happen again?
Yes, that's an excellent question, Max. And you know, one of the challenges goes back to the type of genes that breeders want to use, and you know, they love single dominant genes because they're easy to breed for, and they provide very robust resistance. But unfortunately, those are the absolute worst genes to use, because even though the resistance is robust, they're the type of genes that the pathogens tend to break resistance. So, one strategy is to focus on more horizontal resistance that is multigenic, that may not provide the absolute resistance where there's no symptoms, but provides enough resistance that growers will accept it, and that it will make it difficult for the pathogen to come up with multiple mutations in the same genome to overcome it. So that would be, one way. Or the other way is to, deploy multiple vertical resistance genes at once that are effective, so that once again, the pathogen has to have more than one mutation to break that resistance. But Max, the problem is that we're not able to find enough sources of resistance to this strain. If we could, those would be some of the strategies, that would be used. And to tell you how, I wouldn't say desperate, but interested our processing tomato growers are. Normally, if you just even say the word transgenic in the tomato meetings, they'll throw you out of the room. I'm exaggerating a little, but they don't want any hint of transgenics in their product. And the reason is, of course, because they're on the world market and they want to ship to Europe. And Europe, of course, is anti-GMO. So they want to say that their tomatoes are non-GMO, which they are. But there's somebody in North Carolina who has a transgenic tomato with resistance to some strains of spotted wilt, and they want to test the RB California strain. And the growers supported a little project to do that, and we're providing them the RB strain from California, and they're going to test on these genetically engineered tomatoes to see if they provide resistance. Now, that doesn't mean they'll be, deploying transgenic tomatoes in California anytime soon. But if it works, at least it's something, and maybe that could give us some hints on how to come up with non-transgenic resistance more. So that so there are and then of course going in and using gene editing. Because there's a whole gene cluster of these, of SW5 is located, as you know, Max, in a cluster of R genes, okay? And so there's a number of SW genes in there that are R-like genes that may have useful resistance too. So going back in and SW5B turned out to be the best of the bunch through conventional genetics, but maybe through gene editing, you could come up with some better versions that would recognize RB California, which would be a fun kind of experiment to do. But those are more, basic research projects down the line. So that's what, but until we find these sources of resistance, it's even going to be hard to do that. So then finally, Max, the last thing I would say is what I told the growers, when they got the SW5B varieties in the first place. If you only rely on the varieties, if you don't control thrips, if you don't use sanitation, if you don't make sure that, the transplants are clean coming out of the greenhouse, which they still did that you don't use good then you're going to lead to higher pressure on SW5B, right? Because if you're controlling the thrips, you're using good sanitation, you're keeping the pressure lower, you have a lower probability of coming up with that mutation. And incidentally, that mutation, Max and everybody else, it had to be 2 nucleotides in a row in the one to give that Y residue at 118. Okay, so it wasn't a single nucleotide change. It had to be 2 nucleotides to give that codon for tyrosine, for Y, okay, for the Y amino acid. So think about that. I mean, that had to be a random double mutation that came up. So that just tells you, how many mutants had to be gone through before you came up with that double mutation in that right spot, and then everywhere else you were okay. I mean, that tells you the power of the virus to mutate. It's just incredibly powerful. So if you put more pressure on that virus, it is going to respond the way it did, unfortunately, and it's not the first time it's happened, nor will it be the last. But you raise very good points, Max. But you go to a breeding company and you ask them, hey, how many varieties are you developing with horizontal resistance? I don't think you're going to hear very many. All right, I could be wrong. I hope I'm wrong, but I'm afraid, that's probably not.
Okay. There is one question, Bob, from Ahmad Yusuf Ibrahim. I think he is our master student. It's about the use of resistant varieties also. And you just mentioned recently, just a minutes ago, that he is asking whether is it available, is there many resistant variety available in the market? And if it is, whether it is good to plant them all in one, for example, in one location, so the virus will be, more difficult to develop the resistance, something like that. If the variety, resistant variety is available, meaning.
And that, and that's an excellent question. Unfortunately, we don't have such sources of resistance. In this, when we were inoculating the plants, there was a gene there called SW7, which is another one of those SWs from that R gene cluster. But in the variety that we got that supposedly had SW7, it didn't perform very well. There's another one called SW1, and they're trying to pyramid kind of what you're suggesting, and your thinking is very good. They're trying to pyramid SW1, SW7, and SW5 all together to see if they can get a reasonable level of resistance that would be more robust and harder for the virus to break. So.
Okay. Another question from Naimas. She is an undergrad student in our department. She's asking, since the virus has no, chemical approach to control the virus, like in bacteria, we have a bactericide, like in fungi, we have fungicide. So we really rely on the immune system of the plant, something like that, she said. So is there any approach?
That's correct.
Yeah, is there any approach to induce immune systems in the plants when we don't have the resistant variety? Yeah.
Well, that's an excellent question. Good job. Okay, I think that's a really, really good question. So, there are a few ways that you can trigger the immune system of the plant, okay? And I'll give you 2 examples. So one is called, so many people well, they may or may not know that, when you have a headache, and you take aspirin, right? Aspirin is called salicylic acid, okay? And salicylic acid is a very active compound. If you spray salicylic acid on plants, it triggers the immune system. It turns on defense response genes. There's a whole salicylic acid pathway in plants that is involved in conferring resistance to pathogens like viruses and bacteria and things like that, okay? And there's a commercial product that you can buy from a company. It's called Actigard. It's a mimic of salicylic acid. And you can buy this from the company Syngenta. And you can spray it onto the plant and you can activate the immune system. And there is evidence that it will reduce the symptoms by many pathogens, bacteria, fungi, powdery mildew, it's particularly good, and some viruses. But the amount of protection that you get is debatable, okay? It does provide a beneficial effect. It's not real strong, but it is a positive effect. Now, the second way is through turning on what is called RNA interference or gene silencing, okay? Now, this is a very powerful system that is found in plants, okay? And this system surveils the plant cells for abnormal nucleic acids, okay? Because abnormal nucleic acids are bad news. You don't want abnormal nucleic acids in your body, right? You only want your nucleic acids. So plants, and us too, have developed systems to scan for abnormal nucleic acids, especially RNAs, double-stranded RNAs to be exact. And these, when they're recognized, trigger gene silencing where the plant specifically degrades that RNA, okay? Now, we know that plants recognize plant viruses by double-stranded RNAs and degrade them, okay? And it's a very powerful type of resistance. So the idea is that you trigger that by introducing that double-stranded RNA to the plant to turn, to turn on that very specific RNA degradation pathway. Now, people have tried spraying small double-stranded RNAs on, those will be taken up by cells and they will turn on defense responses, but they're a little harder to engineer. The way that it's been done the most is to make a genetically engineered plant that makes a little double-stranded RNA. You make a hairpin and that gets cleaved out and you make a little double-stranded RNA in the genetically engineered plant that turns on. So that's a double-stranded RNA, let's say for tomato spotted wilt virus, okay? So when that is recognized in that plant, so that when the real tomato spotted wilt virus RNA comes in, the plant is already primed and it degrades that RNA so it never can replicate and infect the plant. This is a very effective strategy that has been shown to work for a number of viruses, including for some Gemini viruses. So in my opinion, that is the most powerful way to turn on the defense system against the specific pathogen, okay, by triggering RNA interference. Right now, the best way is using genetically engineering to trigger that, but many companies are trying to come up with ways to deliver those small RNAs by a spray or some other novel way of introducing them, okay? And the way you're going to do it, think about the vaccine that, we're getting for COVID, okay? I go back to that, all right? Because, the vaccine for COVID-19 is a piece of RNA that has a promoter and then the spike protein gene. And it's delivered into cells by liposomes. So liposomes are little lipids that will fuse to membranes and then introduce their cargo. Okay, so when you get the injection of the vaccine for coronavirus, you're going to get millions and millions of messenger RNAs coated with liposomes that bind to your cells, fuse to your cells, and get taken up into your cells of your body to make antibodies. So it's probably happening in your bloodstream. Okay, so how can we do that for plants? So that's the difference. The plants, there's nothing like an antibodies. That's why I hate the use of the term immune system in plants, okay? I've always hated that. But, you know, the reason they use it is because they can get more money, you know, because if plants have an immune system, they can go to NIH and say, hey, plants have immune systems too, so fund our research. So I don't blame people for using it, but you know, when you say immune system, you usually think of antibodies, right? And of course, plants don't have antibodies, but this powerful gene silencing system, RNA interference, which is found in animals and fungi and all eukaryotes, is a powerful system that we could harness better to make resistant plants, okay? But again, you know, with people being anti-GMO, you know, some of these strategies, you know, just will not be accepted until, you know, and I'm not saying that we should, you know, all be going GMO, but I think if GMOs are, you know, tested carefully and the right way, and they're found to be safe, you know, they should be approved. You know, and like in the US, there are, you know, many transgenic plants that have been approved, including some that are eaten. Soybeans for Roundup, corn for BT, you know, so, and of course, squash for viruses. There's virus-resistant squash genetically that you can buy, that's on the market here. And of course, the famous transgenic papaya in Hawaii that Dennis Gonzalez made. So transgenic have been approved in the US, and we've been eating them for a long time. So we're not asking any country to test them for us, okay? We've done it ourselves, all right? So I'm perfectly convinced that these transgenics are quite safe. And, you know, I personally think we should be more, you know, accepting of that technology. And I think we will be someday. But going back to the original question, that to me is the most powerful way to modify the plant immune system, is to trigger that RNA interference, okay? We know how to do it, we can do it, it is very effective, and you can link multiple little double-stranded RNAs to multiple viruses and make multiple resistance in one transgenic plant, okay? So they've done that in soybeans. And, you know, melons are known for being infected by multiple viruses. In fact, we're dealing with a late season mixed infection of potiviruses, cucumber mosaic, alluvial virus. Oh my God, alluvial virus. So, you know, so you have multiple infections. So by, triggering multiple using multiple dsRNAs to trigger this gene silencing, you could make multi-virus resistant plants. So those are some of the technologies that are out there, you know, and that can be, you know, modified and manipulated. But, you know, we're still not there with that technology.
Okay, so I think there is questions actually when you are explaining about RNA intermediate. This is from Susanti Mugi Lestari. She is a PhD student. Hello, Bob. I just read it. Your presentation was really great. I am Santi. I would like to ask, what do you think about the RNAi to break down the gene in the whitefly involved in the begomovirus transmission? Will it be possible to apply widely in the field? I think you explained just recently.
You know,
yeah.
That is a wonderful question, okay? A wonderful question. So one thing that, you know, we haven't identified, you know, in these persistently transmitted viruses spread by insect vectors is the receptor that takes up the virus in the midgut, which is one of the main areas that the uptake occurs. So, to me, that's one of the holy grails that's left out there to be identified. What are the receptors? Because then if you can identify those receptors, you would then be able to target them with RNA interference and knock out the receptors. You can feed insects double-stranded RNAs through, feeding them sucrose, 15% sucrose, double-stranded RNAs. You can knock out any gene in insects, okay? So RNAi is very effective in insects. They've knocked out eye color, they've knocked out wing formation, they've knocked out many genes in insects. So it is definitely a viable technology. And in fact, Monsanto, now Bayer, had developed a technology where they sprayed small RNAs onto plants and their target was leaf-feeding beetles, like the Colorado potato beetle, chrysomelid beetles that chew on leaves. And they got it to work because the beetles were eating the leaves. The RNAs were pretty stable. They would stay on the leaf. And then it screwed up one of their digestive enzymes or something. It had a pretty high mortality. But the problem was, and still is, how can you apply that on a large scale like you're suggesting and get effective control? And I don't think they solved that. They were trying to apply it in liposomes, all these same things we were just talking about, but they never came up with something. And I believe they dropped that technology, unfortunately. So it's still the issue. We would love to identify the receptor. We have a virus here called curly top virus, which Asti also knows is a Gemini virus that's transmitted by a leafhopper, the beet leafhopper. And it's a problem here on tomato. I didn't talk about it because I didn't have time. And we're trying to sequence the genome of that leafhopper. Well, that's the first problem, because you might think a leafhopper has a small genome. Well, it doesn't. It has a humongous genome. And this has turned out to be a nightmare just to get it sequenced. But the idea would be then to identify candidates for receptors and knock them out with RNAi, and then see if you can knock out virus transmission. And then if you could release insects that you've knocked out the receptor gene, let's assume it's, of course, if it's an important receptor for the insect, you're going to hurt the insect. But let's say it's not. Then you could release those insects out into the wild, and they could compete and mate and, maybe establish, because those insects don't hurt tomato by their feeding. They only hurt tomato by transmitting the virus. You could then, that's the gene drive idea, right? This has been put forth for mosquitoes and dengue fever and things like that, with some success, okay? But you need to identify those genes that are involved in the uptake and transmission, okay? Because I think those are the ones, you really need to target. So your idea is awesome, but we need people to crack that nut, as we would say, of what are the receptors. It's probably endocytosis. I'm almost sure it's some endocytosis, but there's quite a few endocytotic genes in the genome of these, that are involved in that. But I'm sure that's what's involved, and those would be the genes we'd be looking at to knock out and things. So, you young people, there's so much exciting things to do out there. It's and your questions that you're asking are bringing up really good areas where there's just tremendous opportunities for the future. So, don't let this COVID stuff get you down. As a matter of fact, learn from this COVID stuff. It's really interesting as a virologist. I am on the TV watching every night the news shows to, when they have people on, they bring on, very good experts to talk about COVID. And as I say, as a plant virologist, I just find it fascinating. Okay. because we're overlapping in the same stuff. And as I wrote this article, and I hope I'll get the energy to write it as a letter to the editor, and it is, hey, us plant virologists are doing the same stuff, but for a lot less money. And we're doing all of this. RT-PCR, we're sequencing our virus. We have infectious clones. We're trying to knock these genes out. So, we've been doing all this too, but for a heck of a lot less. All right. Not that I'm questioning, of course we should be. The answer is to invest more into science. Okay.
Yeah.
And this science denial stuff that's going on is just so shameful. Climate deniers and vaccine deniers, and what is going on in this world, okay? We're scientists, right? We do things based on testing hypotheses and coming up with conclusions based on results. This world's crazy. And so we need young scientists out there, talking to people, telling people that, we've got to do science-based research and make conclusions based on that. And, it's very dangerous when that's not the case. Okay. So, you young scientists, go out there and, be proactive. One of the things we don't do as scientists is interact with the public enough. And I'm guilty of that. I don't go out. But when my daughter was a kid, we went to her elementary school and gave talk to the kids. When I'm on an airplane, when I used to be, whenever somebody asked me a question, if, I was from UC Davis, or I was usually editing some poor student's thesis, and they said, "Oh my God, what are you doing?" And then they would ask me questions, well, what do you think about transgenic plants? Or what do you think about this? And I would always spend the time to talk to somebody like that to explain, especially transgenic plants, because they never know what a transgenic plant really is. Okay, and then when they hear about it, then they're like, "Wow, well, really, that's not really that bad, is it?" And I'm like, "It really isn't."
There is one question, Bob, about transgenic from students. I think she is undergrad students in the first year, I believe, and
Okay.
she wants you to convince her. This is from Galuh Ayu Cantika Dwitara. Please convince her that the transgenic has no, has no side effect on the human health.
Yeah. My answer to that is that before any transgenic plant gets released for commercialization, it has to go through a series of feeding trials. It has to go through a series of trials where those plants their pollen is looked to see how far it can be released and how, and can it be a source of the transgene. So all of these things have to be done before any transgenic plant would be released for commercialization. And I feel, so in our country, the FDA, the EPA, and the USDA have to approve genetically engineered plants. So they go through human feeding, animal feeding, they go through allergy testing, they go through all of these things. And I believe that once those tests are done and they come through as not having any negative effect, that's, to me, I'm convinced that that's a safe product. It's exactly the same with the vaccine, right? So our FDA has approved the Pfizer vaccine, okay? And they're very soon going to approve the Moderna, and they're both mRNA vaccines, correct? Okay. So when our FDA has done those tests, they've already done, small scale and large scale trials, and they haven't found evidence of negative effects, other than a few allergic responses. To me, that's a safe vaccine. I'll take it tomorrow if I can. Okay. So if the tests are done in that way, I feel very confident that such transgenics are safe. But let me say also, I totally support labeling. So I say if a food has a genetically modified component, it should be labeled. So somebody who really doesn't like it, you know, for whatever reason, they're just against it, you know, and somebody who only likes maybe organic foods might feel that way, then fine, they should be able to see that and make the choice. But I believe that after they've been tested in the way I have said, that they should be allowed, they should be commercialized, and people who want to eat them should be able to eat them.
Great. Okay, now another questions. This is about the detection method using the like test pack you mentioned earlier. This, I think, serology you mentioned.
The Immunostrip.
Yeah, Immunostrip.
Yes.
Yeah.
Yes.
There is
Yes.
a question from Desyanto, our undergrad students in the Department of Plant Protection. He's asking whether the Immunostrip can detect the virus with low concentration.
That's a good question. You know what? No, they're not really sensitive. Okay. So that's one of the negative sides of them. You know, they pretty much, you know, well, I work with bacteria, they get down to about 10 to the 2 colony forming units per milliliter, which isn't that bad. And viruses, I don't know what their limit of detection is exactly, but they're not anywhere near as sensitive as ELISA. They're not anywhere, you know, LAMP is more sensitive than even PCR. So you're only going to use them as diagnostic tools with infected plant tissues. You wouldn't use them to detect the pathogen in seeds or anything like that. Okay, it's pretty much in fresh tissue, tubers, fruits. You know, tomatoes, there's tons of tomato spotted wilt in fruit, for example. So the Immunostrip works great for that. So yeah, they're not that particularly sensitive.
Okay, another question.
But let me ask you this:
All right.
how many of the people in the audience have used an Immunostrip? You know, many of them had exposure to that technology?
No, Bob, because it's difficult to get those materials here. We just do serology, ELISA mostly. Yeah.
Mm-hmm. Okay.
Yeah. Okay, so this is maybe this one you like to answer. I want to ask if the infected plants of infected TYLCV plants can, let's see, sorry, I rephrase again. Andri Saputra asking whether the seed from infected plants infected by TYLCV can born the virus. So whether TYLCV is a seed-borne virus.
Oh, great. You guys are asking good. You guys are asking good questions. So that you're telling me you're thinking. Okay, don't be shy about asking questions. Okay, so this is a great question. All right. So my answer is it's complicated. Okay, and I don't believe TYLCV is seed transmitted at all. Okay, or if it is extremely rare. Okay, now. That being said, there was an article published now, when was it, 2015? Don't quote me on that, but it was in Scientific Reports. It was by a group from Korea, and they claimed that TYLC was seed transmitted, okay? But it was almost all based on PCR, okay? And they never actually took any of the seeds and planted them that, you know, that were supposedly carrying it, and then had plants grow up and get TYLCV. You know, so they never finished the final experiment, convincing experiments, but they claimed based on their data that TYLC was seed transmitted. And unfortunately, this journal published it, because when I read that paper, if I was the reviewer, I would not have accepted it based on what they indicated. So immediately after that paper was published, you know, I start getting phone calls. Oh, is TYLCV seed transmitted? Is that true? Is that true? And I said, you know, it goes against every biological thing we've seen with it, okay? I mean, many of those seeds that we grow in California are grown in Thailand, in places where there's TYLCV, where they make the hybrids. And, you know, theoretically, there's, you know, some of those seeds would have gotten the virus. And then when you plant those seeds somewhere else, you'd see a plant with TYLCV pop up in, you know, in the field somewhere randomly, not, you know, in a place where TYLCV has never been found before. That would be evidence that it came in on the seeds. I've never seen any of that evidence. We've never seen, you know, convincing evidence. And finally, let me just say, in 2019, a group from Spain went back and revisited whether TYLCV is seed transmitted. So as I always tell people, if somebody tells you they're going to revisit your work, that's not a good thing, okay? That means they don't think your work's right, okay? So they went back, and the Spanish group is a very good group, Maria Rojas and et al., and did a very careful analysis. And they got plenty of TYLCV infected plants in Spain to do this experiment to get fruits. The other problem is it's hard to get fruits from TYLCV infected plants. That's the other problem, okay? Because the flowers drop off and it's very hard to get many fruits anyway, okay? So, but Maria Rojas and them were able to do it. And to make a long story short, they came up with, their evidence was that it's not seed transmitted. So they're arguing that, you know, the Koreans, yes, you can detect it by PCR up in the fruit. We know that. But the vascular connections with the seeds break down very early in the production of the seed, okay? So,
All right.
So for the phloem limited TYLCV to move up and get into the fruit and get into the seeds through the phloem is virtually impossible. I think.
Great. Okay, thanks, Bob.
Great question. And
Another, I think one student would like to ask directly. Is it right, Marsha? Are you raising your hand? Marsha Alhanan. Please, Marsha.
Oh, yes, I would like to ask.
Introduce
all,
yourself,
for,
please. Yeah.
Okay, my name is Marsha Alhanan Sifai, and I was part of the Permata Sat program. And first of all, I feel very thank you very much for the lecture today. It's very amazing for me. And I would like to ask, so far, is there we need to perform integrated pest management strategies in order to control the thrips as the vector of this particular disease. So far, what are the integrated pest management that already have been done by the companies or the growers so far? The integrated pest management that have been done. Perhaps those integrated pest, is there any possible to perform the integrated pest management strategies that doesn't involve chemical pesticides? Thank you very much for the opportunity.
Okay, that's an excellent question also. So are there IPM strategies, you know, for thrips that don't involve or aren't solely based on chemicals? Well, so there's two ways to look at that. Now, if there are predatory thrips, biological controls that can be very effective against the thrips that transmit viruses. And there's about 13 species of thrips that do it, and there's probably 5,000 total species. So only a very small number of them. And some thrips are predatory thrips, and they eat other thrips. And in a greenhouse situation, that can be very effective in controlling thrips, is by releasing. You can buy them from these Copert or these companies that commercially sell predators, and predatory thrips are one of the things that you can buy and that you can sell. And in a greenhouse situation, and in Spain, southern Spain, for example, they grow huge amounts plastic house type tomato production in the southern part of Spain. You can see it from the satellite up above the earth. That's how much plastic that they have down there. And they, I hear, are getting very good management of thrips using biocontrol. But I think in large open field situations like we have in California, it's not commercially viable, and I don't know if it would even work to release those predatory thrips. There are other ways to encourage predators, you know, but in our commercial situation, those have not, those are also not economically viable. So for me, that's probably the best non-chemical thrip strategy that I'm aware of. You know, even here, thankfully, we have a winter. The thrips don't like cold temperatures, that's for sure. And that knocks them down quite a bit. But you know what I think? I haven't been able to prove it yet. You know, the thrips fall, you know, they pupate and they fall into the soil to pupate, okay? And then the adults come out. I think, and we know that pupated thrips survive in soil over the winter here. I think some of the emerging adults have the virus from the soil and they start the, you know, because our growers are like, where is it coming from? You tell us that the weeds don't have it, you know, we get rid of all the weeds, we all use all the sanitation, but it still shows up. Where is it coming from? It's not seed transmitted. So how the heck does it show up? And, you know, and I don't know, but you know, and so this, as a matter of fact, this year, we're going to put yellow sticky cards out and we're going to collect, some poor person in the lab has to do this. It has to collect thrips off the yellow sticky cards, and we're going to do RT-PCR. And we're going to see if we can capture any of those emerging thrips early in the spring that are carrying that virus. So back to the, you know, but back to the thrips, you know, there's a soil-borne fungus called Metarhizium that you can spray into soil, and that will infect the thrips in the soil, those pupaean things. So that can also be used more in a greenhouse situation, but to put it on thousands of acres like we grow here with processing tomatoes, it's too expensive. So in protected culture, biological control is a definite option for thrips, and it can be used quite effectively. In open field, in large open field production, there's not, you know, putting out, you know, some growers, they put out big pieces of yellow sticky plastic with, you know, hoping that they're going to capture. I mean, they capture a bunch of insects, but they probably attract more of them because of the yellow color anyway into their areas. So, I mean, you see all these, you know, attempts, you know, to control insects in these ways. And, you know, just like trap crops and stuff like that, it just doesn't reach the level of providing the control necessary to make, you know, farmers convinced it's helping them. And so we, you know, but that doesn't mean those strategies aren't out there and we haven't found them yet, you know.
Okay, thanks. Is that clear, Marsa?
Yes, thank you very much for the question.
Yes,
for the
thank
answers,
you.
I do apologize.
Thank you for asking the question, okay? It's a good
Okay.
question.
Okay, thank you.
Anybody else wants to raise hand? We still have a few minutes. Masih ada lagi pertanyaan apa saja? Okay, Bob, I think we are coming to the end of this lecture. Before we finish this lecture, wait a second. Yeah. On behalf of the department, we would like to give you certificate for our appreciation. We cannot give you anything else, but hopefully through this event, we can, you know, start our collaboration again. And when the pandemic is over, hopefully you will be able to visit us in Indonesia, and just let us know when you are ready to come to Komodo Island. We will guide you, because I would like also to go there. And
Okay,
students
Asti.
probably.
You know what? That's a deal, Asti. Okay, I'm coming. Okay.
Okay, great.
And,
Okay, Bob. Yeah.
okay. Thank you, all wonderful students. It was an honor and a pleasure. Okay. Thank you, Asti.
Yeah, it is a very motivated lecture, Bob. I mean, you don't only share your knowledge and experiences, but you give the students some motivation. And I like the statement you gave in the beginning of this lecture. There is always need for a plant pathologist or plant protection. So this is very important for the students here. And then also another one, everybody need to learn molecular biology. And it is also very important for the students here. Especially at least as tools for diagnosis. I think that's a very good motivation for students in our country here. Okay, Bob,
Exactly.
I think that's it. I think the end of the lecture. Again, thank you very much. Hope we can meet in person sometimes, and give my best regard to Maria.
I will do that, Asti. Thank you again. Please give my regards to your family. Okay.
All right, I will.
Okay.
Thank you.
Bye-bye. Thank you again.
Bye. Thank you.
Bye. Thank you, Prof.
It was a
Thank you,
Okay.
Prof.
Thank
So,
you, Bob, for your sharing knowledge.
my pleasure, everybody. My pleasure. Thank you again.
Thank you, Bob.